Binduprava Sahu. Welcome you all to SNEB'S Biology. In the previous video, we have started discussing the various models which explain the structure and composition of the plasma membrane. So today we are going to discuss about the fourth model, that is the Fluid Mosaic Model. Okay, and this Fluid Mosaic Model was proposed by Singer and Nicolson in the year of 1972. Okay, so what Singer and Nicolson did, they just modified, interpreted the previous models which was given by Robertson as well as Danielli-Davson. Okay, so what we have discussed, that the first point, that the plasma membrane is a phospholipid bilayer with protein. Okay, so there is protein and there is lipid bilayer, right? Yeah. So considering the protein, Singer and Nicolson interpreted that proteins are of two types. First one is the intrinsic protein, and the other one is the extrinsic protein. Okay, so what is intrinsic protein? The intrinsic protein is the protein which remain completely or partially embedded in the lipid bilayer. Okay, lipid bilayer. Clear? And what are extrinsic protein? So these are the protein which are outer to the lipid bilayer. That means they are present attached to the head of the lipid. Attached to the head of lipid. Okay, clear? So if we consider the structure, how it will look? This is the head and tail of the lipid bilayer. Okay, this is the head part, this is the tail part. Clear? And in some lipid present the phosphorus group. That kind of lipid is known as phospholipid. Okay, so attached to the head present the extrinsic protein. Okay, so these are the proteins which are extrinsic. Clear? And there are some protein which are partially or completely embedded in the lipid bilayer. Okay, this is the completely embedded protein, this is the partially embedded protein, and both these proteins are called as the intrinsic protein. Okay, these are the intrinsic protein. Clear? So these intrinsic proteins are also called as the integral protein. Okay, what are these? These are the integral proteins. Clear? Now come to the second point. So the next point is about the phospholipids. Okay, so what phospholipids are? Phospholipids, I have already told, that lipid which has phosphate group at the head part. So consider a lipid molecule. This is the head and this is the tail. Okay, and this head is hydrophilic in nature, whereas the two tails are hydrophobic in nature. Philia means what? Loving. Philic means loving. They are loving towards water. Okay, and this is hydrophobic. They fear from water. That means they are water insoluble. So hydrophilic part is also called as the polar part, polar head. Here this is the polar head, whereas these are the non-polar heads. Clear? Now, so as lipid is both hydrophilic as well as hydrophobic, so it is amphipathic in nature. Amphipathic in nature. Amphipathic means what? Both hydrophilic as well as hydrophobic. So this was already given by our Danielli and Davson. So what new things Singer and Nicolson added? They told that this lipid molecule shows two types of movement. Okay. There are two types of movement. The first one is transmembrane movement, whereas the other one is flip-flop movement. Okay. First one is transmembrane movement and the other one is flip-flop movement. So consider the bilayer structure. These are lipids. Okay. This is the tail part. Clear? And there are some proteins attached to the lipid. Okay. So these are some extrinsic proteins. Yeah. Whereas this is the intrinsic protein. Okay. Let me extend the drawing. Another. Okay. So let's number these lipids. This is the lipid number 1, 2, 3, 4, 5 and 6 in the first layer. Okay. And in the second layer, let us name them A, B, C, D, E, F and G. Okay. So according to the transmembrane movement, the movement of lipid is within the layer, within the same layer. Okay. What it means? Within the same layer. So consider this structure. So this is the lipid number 2 and this is the lipid number 5. So if it is showing transmembrane movement, then what will happen? Then 5 will come towards 2, whereas 2 will come towards 5. So what will be the numbering? This will be 5 and this will be 2. The movement is only within a single layer, a same layer. Okay. So if we consider the movement within second layer, so let us see that C and E, they are moving. Okay. So E is moving towards C, whereas C will move towards E. Okay. So this will be the lipid E, whereas this will be the lipid C. So this type of movement is called the transmembrane movement. Okay. Now, next the flip-flop movement. Okay. So let us restore our structure. This is the number 2 and this is number 5. Okay. So if it is showing flip-flop movement, then the movement will be between the lipid bilayer. Okay. So this is the lipid number 3 and this is the lipid D. So D will move towards 3, position of the 3, whereas 3 will move towards the position of the D. So this will be our D lipid, whereas this will be the lipid number 3. Okay. So this type of movement, the movement is between the lipid bilayer. Okay. So that kind of movement is known as flip-flop movement. Clear? So these are the two general characteristics of the plasma membrane, which was proposed by Singer and Nicolson. Okay. So this is all about the fluid mosaic model. There is another thing which they have added, that they described this fluid mosaic model as the icebergs of proteins in the sea of lipid. Okay. What they said, that the plasma membrane looks like icebergs of proteins in the sea of lipid. Okay. So what are icebergs? We all know that icebergs are the chunks of ice. Okay. So they described that these protein molecules are like icebergs which slide over the sea of lipid. Okay, within the lipid bilayer, means that these protein molecules are able to slide over the lipid bilayer. Okay, clear? So this thing is explaining the fluidity of the plasma membrane. Okay, or the dynamic nature of the plasma membrane. So plasma membrane is also called the quasi-fluid structure or membrane. So what do you mean by quasi-fluid? It means gel-like. Okay, it is neither too liquidy or too solid. Okay, it is gel-like, so that it provides a suitable environment for the proteins to slide over the lipid bilayer. Clear? So now let's discuss about the dynamic nature of the membrane. Okay. So considering the property of the plasma membrane, the first property is saying that both fat soluble as well as water soluble substances can pass through the plasma membrane. Okay. So let's draw the diagram according to Nicolson and Singer. Okay. So these are the lipid bilayer. Okay. Then there is present the proteins. These are the proteins. So we all know that the head of lipid is polar. Okay. And the tail is non-polar or hydrophobic. So this environment, okay, so this environment is suitable for lipid to be transported, right? So if a fat soluble substance is coming, then it can easily pass through the plasma membrane. But what about a substance which is water soluble? Okay, so consider a water soluble substance here. So it will reach till this layer, but this layer which is completely filled with lipid, it will not get soluble with it, right? So there comes the action of this integral protein. Okay, the intrinsic protein. So this protein will act as a channel protein. Okay, and this molecule will bind with this protein and it will then transported easily to the other part of the cell. Okay, clear? So if we consider the various parts, the lower part will be facing towards the cytoplasm. Okay, so this is known as the C phase. And the outer part is the exoplasm. So this is known as the E phase. To transfer the water-soluble substances from E phase to the C phase, it will need a channel protein, okay, which is also a integral protein or intrinsic protein. So the first point is satisfied that both fat-soluble as well as water-soluble substances can pass from one lipid layer to the other. Okay. Now, come to the second property of the plasma membrane. It is said that the protein to lipid ratio is variable in the plasma membrane. Okay. So, the protein, consider this structure. Here, let us take this much structure. Clear? And let's draw some other proteins also. There are some proteins. So, in this part, how many proteins are there? Protein, there are 1, 2, 3 and 4. Okay, there are 4 proteins. And how many phospholipids? There are 1, 2, 3, 4, 5, 6. 6 phospholipids. Okay. So, let's equate the number of proteins. Let's draw another 2 proteins here and here. Okay. So, the number of proteins is also 6. So, if the plasma membrane is showing any transmembrane movement or flip-flop movement, so what will happen? First, consider the flip-flop movement. Okay. So, in flip-flop movement, what happens? Now, this lipid will come towards this one. Okay. This is coming towards this one and this will go to this one. So, the total number of lipid will remain same. But what about the transmembrane movement? Okay. So, consider this lipid and this lipid. They are moving towards their respective positions, right? So, at one position, this position as well as this position will remain vacant. So, the number of lipid will what? Will be, the total number of phospholipid will be 6 minus 2, that is 4. Okay, clear? So, the ratio will vary. Considering again the flip-flop movement, this is one lipid, this is another lipid, and at this position there will be vacancy here as well as here, right? So, the number of lipid will also be 4. Clear? So, it is proved experimentally that the protein to phospholipid ratio will vary. Okay. Then, the third point is about the fluidy nature of the plasma membrane. As I have already discussed, to describe that, Singer and Nicolson told that our model, in our model, the proteins and lipids are arranged in such a manner that the proteins are compared with the icebergs and the lipids are comparable with the sea. Okay, clear? And within the lipid bilayer, the protein keep floating. Clear? So, this is all about the 3 points and the fluid mosaic model that was proposed by Singer and Nicolson. I hope you all understand what I have taught, right? So, if you have any doubt, you can go to the comment box and comment, ask me any question, and don't forget to subscribe to the channel, press the bell icon next to the subscription button so that you can get all the updates from us. If you are liking my video, do like and share with others so that you will all help in spreading knowledge. Thank you.